Okay, well, welcome to the return of the Spotlight Seminar series. And this month's meeting is going to focus on mosquito ecology and arbovirus studies from my group. Current research focuses on the influence of lethal and non-lethal effects of larval predation and exposure to insecticides and phenotypic traits of adult mosquitoes. In particular, most of our model systems work with Aedes aegypti. That's not always the case, but right now it is for the last couple of years. We'll also be speaking about some projects that we have on the use of fluorescent powder markers in Aedes aegypti and how they may be used as tools to further our understanding of Aedes aegypti biology. And Melissa Dulcey, she'll be presenting on the role of blood meal source, and that is what type of host a mosquito feeds on, and how that blood meal source may influence the midgut microbiome and mosquito and arbovirus infection in Aedes aegypti. Next. So most of the research in my group focuses on invasive mosquito species, especially those that occupy container habitats such as Aedes aegypti and Aedes albopictus. These make for really nice model systems in terms of manipulation in the field and the laboratory system, especially when much of our interest focuses on ecological interactions that are occurring during the larval stages. So it's relatively easy to manipulate buckets or containers of water at different temperatures or exposure to different stressors. And these invasive mosquitoes that occupy containers, they're some of the most important transmitters of arbovirus, especially those that we're seeing emerging in the last 10 years. My group addresses questions usually with ecological themes regarding the ability of mosquitoes to transmit arboviruses. So that includes measurements on the survival of the adult mosquitoes, their propensity to feed, the length of time, their reproduction, as well as aspects of their interactions with the viruses, what we might call vector competence, and comparisons of viral load and other aspects of mosquito fitness. And on the right is a diagram that just shows sort of the process in which an adult mosquito imbibes a viremic blood meal, and there's barriers to infection for that arbovirus in the midgut, going beyond the midgut, incubation time before the mosquito becomes infectious, and then a time in which the mosquito is infectious and can potentially affect other hosts through biting. We study a number of different ecological processes that influence all these stages of the progression and state of infection. Next slide. So this is the lineup of the outline of presentations today. Shawna will be talking about lethal and non-lethal effects of toxorhynchides predation on Aedes aegypti. She's been working for years on this system. All of her experiments are completed. She's done a great job with them. She's already published a couple of papers on this topic, and so it should be a good talk. Deanna is working on our fluorescent powder markers as tools to understand the biology of Aedes aegypti. She's published 3 papers just over the last year on different aspects of fluorescent powders and how it influences mating and survival and capture rates of mosquitoes in a semi-field environment. Abdullah, he's working on insect growth regulators, in particular pyroproxyphene, and how that may interact with other sources of mortality in nature, such as predation, and influence the phenotypic traits of adult Aedes aegypti. This is an interesting ecological system. It's also an interesting system from a practical point of view in terms of thinking of combinations of control agents. Melissa has been doing work on a project that's been funded through UF on looking at vertebrate source of blood meals and how that may alter the midgut microbiome and arbovirus infection She's doing a series of very interesting experiments. Bob Aldridge recently came down and finished a very large experiment looking at the interactions of sublethal insecticide exposure to Aedes aegypti and subsequent influence on infection with dengue virus. Then the second part of that is to look at mosquitoes that are infected and how they might be more or less susceptible to insecticide sensitivity in the adult stage. So a number of different sort of themes showing up. And with that, if you could turn to the next slide. I think we're ready for our student presentation, starting with Shawna.
Okay, let's see here. Screen. There we go. Okay. Well, good afternoon, everybody. My name is Shawna Bellamy. Most of you know me, but if I have not met you before, hello, welcome to FMEL. And I'm going to be speaking on the role of predation in determining the traits of a mosquito and its infection with Zika virus. So, a little bit of background. When examining predator-prey interactions, typically most research looks at the directly lethal effects. So, how quickly can this predator kill the prey, and how many can they kill? However, predation can also have some additional non-lethal effects that can cause some significant changes in prey behavior, morphology, physiology, and life history traits. And as mosquitoes tend to be major vectors of human disease, it was logical to examine the potential impacts of non-lethal predation on vector-virus interactions. In particular, for this experiment, we focused on the possible effects on the potential for viral infection, dissemination, and transmission of Zika virus. We hypothesized that predation stress would result in a change in the rate of infection, disseminate infection, and the potential transmission of Zika virus within Aedes aegypti mosquitoes. So, the experimental subjects. We have Toxorhynchites rutilus over here, a predatory mosquito. We have Aedes aegypti on the right, so our vector. And then we have Zika virus, so our pathogen. For this experiment, we hatched Aedes aegypti larvae of the Key West strain from Florida, and then we counted those larvae into groups of 150 mosquitoes. Each one of those groups was assigned to a specific treatment group. For this experiment, there were 3 treatments. There was control, removal, and predator, and each one of those treatments had 5 replicates. For the predator pans, I counted them daily and recorded the number of prey that had been killed or consumed. And that number was used to calculate the daily estimate of mortality. So the total number that were dead from the pan or missing from the pan were averaged across all replicates. Then the removal pans actually used that daily estimate of mortality as the number to randomly select from within the removal pans and remove. Survivorship to adulthood, immature development time, wing length, infection, dissemination, and potential for transmission. So the positive saliva samples were all recorded. So, as I mentioned, groups of 150 larvae were placed into these pans, and these pans were placed into the bioreum. The pans were checked daily for pupation, and all pupae that were found were placed into these snap cap vial tubes, and then the tops were stuffed with cotton, and tubes were placed back into the bioreum to allow the adults to emerge. And upon emergence, the adults were sexed, and then they were released into mating cages. The age range for these cages was about 6 days, and they were provided with sugar water and placed back into the bioreum. Yeah. At 7 to 12 days post the first female release into the cage, females and males were separated and transferred to BSL-2 cages and then moved into the biosafety level 2 lab. Time to pupation, the number that pupated each day, and the sex of the emerged adults and the number of adults that actually emerged were all recorded on a daily basis. After being transferred to the BSL-2 incubator, the females were offered a Zika-infected blood meal. Females that remained unfed were later offered an additional attempt to feed. The females were allowed to feed for approximately 1 hour, and then after feeding were sorted into groups of fed, unfed, partially fed, and dead mosquitoes. The fed and unfed mosquitoes were placed into separate cages so that the unfed could have a second attempt to feed, whereas the partially fed and dead mosquitoes were discarded and frozen in tubes to be used for wing length measurements. At 11 days post blood meal, the fed mosquitoes were sampled via capillary assay, the legs and wings were removed, and the wings were mounted onto slides. The legs, bodies, and saliva were all placed separately into -80°C freezers in incomplete media. And then RNA was extracted from the samples using a QIAamp viral RNA mini kit, and QRT-PCR was run to visualize the numbers of positive and negative samples for each of the treatment groups. Wings were then photographed and measured, and the data was analyzed using SAS. In order to generate the viral load that we used for this experiment, Zika virus was inoculated into African green monkey kidney cells and then allowed to grow for 6 days. Then an aliquot of the virus was combined with bovine blood and ATP and then offered to the mosquitoes via the artificial membrane feeding system. Pre and post blood meal aliquots were taken and stored at -80°C. Manova indicated that there was actually a significant overall effect of treatment on the life history traits of, Manova indicated that there was a significant overall effect of treatment. Survival to adulthood contributed the most. Development time was contributed the next most, and wing length contributed the least. Pairwise tests showed that there were significant differences between all comparisons, except for removal versus predator. This figure right here indicates female survival. The differences in letters show significant differences between the treatments, and the numbers in parentheses next to the letters are the total number of samples for that treatment. And as you can see, female survival was significantly higher in control compared to removal and predator, and there was no significant difference between predator and removal treatments. Development time for removal and predator was significantly quicker compared to control, but there was no significant difference due to treatment in wing length. Additionally, we found no significant difference due to treatment on body titer or leg titer. As you can see, on the right-hand side of the table, I have the mean titers and the significant errors. All of the infection titers fell within about 0.5 of each other, and all of the dissemination titers fell within about 0.5 as well. There was no significant effect of treatment on the susceptibility of Aedes aegypti to infection with Zika virus. The numbers on the right, again, are the total number of samples tested. Additionally, there was no significant effect of treatment on disseminate infection. So the proportion of the samples of the legs that were positive. And we found no significant effect of treatment on transmission potential, so the proportion of positive saliva samples. So non-lethal effects seem to have no real significant impact on viral infection, disseminate infection, or viral titer of Zika virus within Aedes aegypti mosquitoes. This, combined with results of previous research, such as Barry in my previous paper indicating that there was a significant decrease in adult lifespan of survivors of predation stress, indicates that the effects of predators on arboviral transmission may largely be mediated through alterations in life history traits as opposed to direct effects on the vector pathogen interaction. And these results may actually have some interesting implications for mosquito and mosquito-borne disease control programs, particularly those that wish to use biological control agents. The lack of significant effect of predation on vector-virus interactions indicates that a biological control agent would likely result in a minimal risk of, or a minimal increased risk of viral transmission among surviving vectors. And when added to the significant decrease in adult lifespan experienced by survivors of predation, the use of predators for mosquito control might become a more safe alternative to use or overuse of chemical larvicides. And these alternatives may become necessary sooner rather than later, considering we've seen more and more insecticide resistance, and there's also a potential increase in viral susceptibility after sublethal insecticide stress. Future studies should actually look at the impacts of non-lethal predation in different environments and different environmental conditions. So to assess the value of predator-based control strategies for mosquito control programs. Additionally, interactions between predation stress, sublethal insecticide stress, environmental conditions, and pathogen infection should all be further explored. Any comments, questions, or concerns?
We have time
Sorry,
for questions.
how's your writing coming?
It's going. I had to tear apart the fertility and fecundity data analysis and redo it because I found an error in the analysis. So I'm going back through all of it and tearing it apart. The paper for the viral one, we just actually recently submitted it to the Journal of Medical Entomology. So that's 2 chapters potentially published.
Good deal.
Let me know if I should stop sharing screen.
Yeah, go ahead, Barry.
Yes, please.
Okay.
So should I continue? Yes. Or I don't know if there is any question for Shawna.
Does not sound like it.
Okay, so I will share my screen. So everyone can see it? Okay, great. So, well, as Barry shared before, I have been working in a project that relates different impacts or is studying different impacts of fluorescent powders on Aedes aegypti. And we want to use these for, like, at the end, in a practical approach, trying to create a device that will detect and mark females or mosquitoes in the environment. So I will not go like deep into all the experiments, but I will share a little bit like what I have been working in. So we thought about creating this device that will be able to capture mosquitoes, then detect if they are marked or not. Oh, sorry. Detect if they are marked or not, then mark them and release them. So these can have different potential applications in the future. So this is just one of those potential uses that we can, for example, have different traps in the environment. Each of them they can collect, then mark and release different color mosquitoes, and then we can follow them in the field and have information about their biology, dispersal, structure of the population, and different type of interesting questions. But I think this is a very flexible system, so then we can use it in different applications. So before we start all the experiments, we evaluate different type of markers, different way of put them on the mosquito, like external, internal, and different ways. But at the end, we decide to move forward with fluorescent powders. And because of different reasons that I will not go very deep into that, and also we thought, okay, we want to use them in a field like with potential different practical different applications in the field. We wanted to make sure that they would not affect very important mosquito traits, and also we wanted to evaluate other practical questions. So we were like we go like through these different topics. So we evaluate like the impact of the powders in the survival, in the host seeking response, flight performance, blood feeding, copulation, and also other practical things like, for example, are we able to detect them in the environment for several days, or are they being transferred between a marked and unmarked mosquito, or are we able to see more than one powder in the same mosquito? So for all of the experiments, I use a very simple application method. I put the mosquitoes inside a paper cup, and then with the syringe, I place the powder on top of them. Then I use different sources of UV light, mostly like flashlights, and microscope, my compound or stereo microscope, depending on the experiment. And then for having like a more objective way of giving like a score system of the presence of the powder, I use a system of 5 maximum points that they were like, for example, if the mosquito has the powder in the legs and in the wings, then it's like a 2. And then if it is legs, wings, and abdomen, it's like a 3, and so on. So we grouped together, like it was 9 experiments. So we grouped them in groups of 3. And this is the first 3 experiments that we did. They are summarized in this paper. And in this one, what we did was like, we were interested to analyze different brands and different series within the same brand of fluorescent powders to choose which one was the one that we saw the best performance, and then continue working with those. So we used some powders that they are available in the market and very like well known already, but also we use different ones. Some of them that they were never been used before, and even like, for example, in Dayglow, we use a different type of series that it was like new series and it was having been tested before. So in these 3 experiments, we test like the detection of the powders in different conditions during at least 30 days. We evaluate their effect on survival and their recapture using a lured trap that is also like an indirect way of measuring the host seeking response, because the trap that we used was lured with a BG lured that resembles like the host odor. So these are the main results. So at the end, we were able to detect all the powders from day 1 to day 30. So all of them, they were detectable. We use in control environment inside the bioroom, but we also use it in the field. So in all of the powders that we tested, we were able to see them for like 30 days. None of them we detect like an effect on the survival in any of the powders that they were evaluated. And also in the part of the host seeking response, even though most of them we have like high recapture rates with the marked females, they did show a significant difference between the unmarked female, only with one of the powders that it was like the Day Glow Eco Series one. This one we didn't found any difference. So that one actually were like the powders that we continue using for the next of like the rest of the experiments, the other 6 experiments. So it's interesting because this type of powder has some difference between like the regular powders. Most of the use before, like all the powders that have been used in the past, they have a lot of chemical compounds and they have formaldehyde. So these ones, they are like a new series and they are like ecological, that is eco series. So they don't have like a lot of like the regular chemicals that are present in other powders, including formaldehyde. And also they have like a very small particle size and a low specific gravity. So the rest, like the next 3 experiments, they are summarizing this one. And in these experiments, what we tested was like using all different 5 colors of Day Glow Eco Series powders. And we test the effect of those powders in survival, but this time, instead of using only females of one age, we use like 3 cohorts or 3 age groups. We also tested like their effect on the blood feeding success and also on their flight speed using a flight meal. Again, we didn't found effects of the powder on the survival in any of the cohorts. The blood feeding success actually was good. Some of them, like in two occasions, it was like a little bit decreased in comparison to the unmarked female, but all of them, they had like good blood feeding. And the speed was not affected by the presence of the powder using this system that was like a third flight speed. Then the next 3 experiments, they are summarized here, and they were like one experiment related to the effect of the powders on the copulation aspects of Aedes aegypti, and 2 practical things. If we were able to use multi-marking approach, that means like using more than one powder in the same female, if we were able to see like different colors, and also if they were going to be transferred between an unmarked or marked mosquito. So the main results, I will not go very deep into the effect on mating because actually that's the topic that I will present on Thursday on the symposium, because that is like a long experiment. So I will not go very deep, but just to summarize, we didn't found effects of the powders on the different aspects of the mating that we evaluate. It was very interesting because as we like, it's very logical to presume we did found some transfer of powders between mark or male or female to an unmarked male or female. And these yellow, like orange dots in the figure on the left is like the places that we usually saw the presence of the powders and the transference. So we were also interested, like we were thinking, okay, we presume that the transference between powders will occur during copulation, but we were curious if that transference will also occur between same-sex individuals. So we tested from male to male and female to female under different conditions, crowded and not crowded. So we found only transference between males and both in crowded and not crowded conditions. And we were able to record this video in which, well, the ones that are in the video, they're both males. And actually it was interesting because as we can see in the video, well, watching the video, I don't know. You can see that both are like having these interactions that are actually similar to the one that the male has with the female. And those interactions, they were very similar to actually the places where we saw the transference of the marker from male to male. It was like the same places that we found from male to female where they are copulating. And it was independent of density. Yeah, it was more like a behavior transference. And then in the part of the multi-marking, it was very interesting. We were able to, we applied like 2, then even like in one of the experiments, we use only 2 and we evaluate different combinations. And in another experiment, we did like multiple combinations using 2, 3, 4, and 5 powders in the same female. And at the end, like the main findings are that the one here with the eye that doesn't have like a line cross on the left, these ones are like the combinations that we were able to tell apart, and the one that is the eye with the line cross are the combinations that we were not able to tell apart. Mostly, as you can see in the picture at the right top, when you combine yellow and green, at least from the human eye, it's very hard to tell apart between those. So that combination, it was not very good, and we were not able to even tell like the difference. And also when using violet, we have this problem because since Aedes aegypti have these scales that are white, when you use UV light and you want to like check the powders, then that part of the white scales, they also look violet. So it was hard to tell between the scales and the powder by itself. So that every combination that used violet was not also very good. And right now I am working in the part of the development of the detecting and marking device. We have been working in that like since June, and it has been very fun and with a lot of challenges. And now we have like a basic prototype that we are still working on. So the main idea in here is like that they will be trapped. Then there will be like a detecting system that will be using a photo. So they will take a picture of the mosquitoes that are trapped. Then they will be moved to another chamber that will be like the chamber 4 marking, and we will use a device to automatically mark them and then release them. But this is under construction, and then I will share more updates in the future. And that's basically it. These are like, well, all the people that has been involved with the project. And yep, any comments and any questions are welcome.
All right, Kenneth, no questions for us.
I got a question.
Oh yes, please.
Oh, that was a very good presentation. I enjoyed it quite a lot. Actually, it's not a question; it's more a comment. This is a wonderful research project. I thoroughly enjoyed it, and but I think you should be prepared for if you're presenting at a place like a mosquito control organization such as FMCA or AMCA. There may be some questions that ask you about the ethical drawbacks of and then re-releasing female, even though you've captured them. Like, what is the ethical kind of conundrum there if you've captured a female that could potentially transmit pathogens, and then you re-release it? You know, be prepared to address that. So.
Yes, it is a good point. Actually, I never thought about it. I was like more focused on the thing that, like, in comparison between capture them and, or actually no, rear them in the lab, then mark them and release them. You have some like bigger concerns in there, like in comparison between like using field already there mosquitoes. But you're right. Yes, some people can say, hey, if you already collected, what are you going to release it?
What, like, why are you releasing it? You could kill it right now. Kill it. and that's why I'm saying, like, just be prepared for that. There may be some pushback.
Yes, and actually, I didn't well too deep into this because it's like a restricted time. But actually, I think we can also use another approach. We can also use that type of device for capture them, detect them, and then leave them inside a trap without releasing them. That can be an approach too. But obviously, there is other ways that we can use it that has like that will answer different questions. But you're totally right. I will think about that potential question. Thank you.
Potential concern, yeah. Do you ever capture any males in your trap as well, or is there a guideline for that?
Yes, actually, when we did some of the host seeking experiment in the roundhouse, that it was like a semi-field environment, we released sometimes males and females, and we capture more females because we are using a lure that will attract females. But it's interesting. Sometimes I guess the males are around the females, and they will just be sucked by the trap, because once they are like near the trap, there is a fan that will suck them. So we have captured males, but yeah, in less proportion.
Right. This seems like actually a very nice trap for sterile insect technique, mark release recapture experiments. For mark release recapture and sterile insect technique, they mark males and then they release them and recapture them over a period of time from a certain distance away from the release site. And I know that several programs actually do that. Mosquito Mate is one of them. They do a release with pyroproxyfen marked individuals, as well as Wolbachia infected males. So I could see a broad spectrum of applications in this technology. So thank you.
Thank you.
Hello, can everyone see my screen? All right, cool, 1 second. All right, my name is Abdullah Alomar, and I am a PhD student in Alto lab. My research is focusing on the interactive effect of insect growth regulator and environmental stressors on mosquito traits and Zika virus infection. So as we all know that mosquitoes are one of the deadliest animal in the world because of their ability to spread pathogens to human and animals. And since there is no effective vaccine, protecting human from these pathogens has been primarily achieved by controlling mosquitoes population using pesticides. Insect growth regulators are a common group of pesticides that used to control mosquitoes during their larval stages. So there is the use of insect growth regulator has potential to interact with environmental stressors, for example, radiation and temperature, to affect mosquitoes population size and traits and pathogen infection. Pyroproxyfen is a common insect growth regulator that show effectiveness against mosquitoes and other public health insects at very low concentrations. So this feature allow pyroproxyfen to be utilized in auto-dissemination technique. Auto-dissemination technique of pyroproxyfen is a promising technique for mosquito control, where pyroproxyfen can be mechanically disseminated to the new oviposition site to inhibit mosquito from emergence to adult stages by females that previously visited pyroproxyfen treated station or mated with contaminated males. So this unique mechanism of action of pyroproxyfen that inhibit mosquitoes in emergence to adult stage and don't target the larval stage enable pyroproxyfen to take advantage of other source of larval mortality in nature, like predation, to limit the size of mosquito, to limit the mosquito population size. Great. So taxolin kinase, it's wait. Do you guys see? So taxolin kinase, it's a natural predator that consumes mosquito prey at larval stage. It's considered as a biological control agent, and the adult of this species does not transmit pathogens, so there is no risk of disease transmission by using this species. So the first objective of my research was to determine the Aedes aegypti responses to lethal and non-lethal effect of predation and insect growth regulator Previous studies observed that the exposure to predators during larval stages induced non-lethal effect on survival, as Shawna mentioned, reducing the adult lifespan. And also exposure, the larval exposure to pyropaxfen also found to induce some non-lethal effect on Aedes aegypti. So to determine, in my experiment, we determined the interactive effect between predators, predation, and pyropaxfen. We measured the effects of exposure to pyropaxfen and predator, either alone or in combination, during the larval stages on adult survival to adulthood, and also size and also lifespan. As you can see in this figure, the combined effect of IGR, pyropaxfen, and predators heavily lowered the Aedes aegypti survivalship to adulthood more than the independent effect of IGR or predator alone. And I will talk more about the rest of this result of this experiment in the upcoming symposium. Another experiment that I have conducted and during my PhD was to determine the effective exposure to pyropaxfen and predation on Zika virus infection and transmission in Aedes aegypti. So as I mentioned earlier, that larval exposure to predator can alter the traits of adult mosquitoes, and also the exposure to the pyropaxfen have found to damage larval midgut cells. So these observations suggest that exposure to pyropaxfen and predator during the immature stages can have non-lethal effects on adult, which may alter their immunity and susceptibility to Zika virus infection and transmission, as observed with other insecticide. So to determine the effective exposure to pyropaxfen and predation, we conduct a series of experimental manipulations to determine the exposure effect on mosquitoes and Zika virus infection. So as you can see in this diagram, we have 6 treatments. The first one is the control, which had just Aedes aegypti larvae. And the second treatment is the pyropaxfen, which has a single low concentration of pyropaxfen. And the third one is pyropaxfen plus predator treatment, which has a low concentration of pyropaxfen plus a single predatory larva of Taxarenchides. And the fourth one is the pyropaxfen plus a predator removal. And the fifth one is the predator treatment. This treatment has just a single larva of Taxarenchides. And the last one is the predator removal. So with regard to these removals, in the treatment that contain predator, for example, pyropaxfen plus predator and predator treatment, the number of consumed Aedes aegypti prey was counted, delayed, and averaged across all the replicates as a measurement of the daily predation rate. And then this number was removed from the pyropaxfen plus predator removal and predator removal treatment, respectively. The reason for doing this is that we want to ensure that the non-lethal effects on Aedes aegypti traits reflecting the absence of predator stress, but including density reduction, since the density reduction has been found to alter the traits of Aedes aegypti. And then when the adult emerge, we will expose the adult to Zika virus blood meal, and 15 days post-infection, mosquitoes were dissected, their legs and a single wings were removed from the rest of the body, and saliva were collected using capillary tube to determine the infection. The result of this experiment revealed that adult mosquitoes exposed to Taxarenchides predator during the larval stages had lower susceptibility to Zika virus infection than other treatments. However, the significant differences were observed only between the predator and pyropaxfen treatment. Another environmental stress that may interact with pyroproxfen is the temperature. So in this objective, I'm interested in determining the interactive effect of larval exposure to pyroproxfen and temperature on Aedes aegypti factor competency for Zika virus. Temperature has been found to alter pyroproxfen effect on Aedes aegypti, and also temperature has been found to alter Aedes aegypti factor competency for Zika virus. So there is a high potential between pyroproxfen and temperature in altering Aedes aegypti factor competency for Zika virus. So in this experiment, we have exposed larval mosquitoes to pyroproxfen under two different temperatures, high and low. And then surviving adults were exposed to feed on infectious blood meal containing Zika virus. And then after 14 days of incubation of the Zika virus, mosquitoes were allowed to feed on chitonic, I think, chitonic Q paper. I don't know how to say the name, chitonic or chitonic Q paper to determine the saliva infection. And then mosquitoes were dissected as we did in the previous experiment. The data of this experiment are still in the progress, and this is what I have for today. And thank you very much. All right, stop sharing.
Great, thank you for that talk, Abdullah. There's time for questions.
I just have more of a comment on the presentation. If you give a talk like this at a professional meeting or anything along those lines, something you might want to consider is breaking up that figure slide a little bit, because that was a lot of data to look at all at once.
Yeah, this is a good comment, but you know, as this seminar, it's not kind of go deep on the experiment. So
yeah,
I just want to
mention it for when you do some professional meetings, because there's only so much data your audience can take in before they kind of stare at you dazed, like,
Yeah, that's very nice comment. Thank you.
But good job.
Appreciate it. Any questions?
Well
all right, no, thank you, Bob. No question, thank you.
Okay, I think we're ready for our next speaker.
All right, I'll start sharing my screen. Okay. All right. Oh, geez. Hi, everyone. I'm Melissa Dulcey. I am a One Health PhD student with public health and working in the Alto lab, as he had mentioned, on looking at different blood meals that an Aedes aegypti mosquito might take, and that influence on the midgut microbiome and possibly affecting the susceptibility to arboviral infections. So to jump right back into some background, first of all, the microbiome. The definition of a microbiome is a community of microorganisms that inhabit a particular environment. And this can be made up of bacteria, viruses, fungi, but to be honest, bacteria has gotten most of the attention in research. And they vary, the microbiome generally varies between different environments. For instance, in humans, you can have a different microbiome in the mouth versus the gut versus the skin. And the same is true for mosquitoes or other insects as well. And again, in the mosquito, most of the attention has gone to the microbial content of the midgut, as this is the first area in which viruses or pathogens coming into the mosquito really have their first contact with the mosquito itself. And so there are many factors that can contribute to the changes of the microbiome in the midgut of the mosquito. some of them being geographic, species of mosquito, as well as sex, developmental stage, and diet. And diet's a really important factor in this, as it has the ability to change the midgut microbiome very quickly. And previously it was thought that we only really saw this change when a mosquito fed off of sugar or nectar versus blood itself. But now we're appreciating a little bit more about some of the changes that occur if a mosquito feeds on different species. So this was first discovered in Ixodes pacificus, which is a tick that is a vector for Lyme disease, where when it was fed lizard blood versus mouse blood, it had very different microbiome. And then this later was seen in Aedes aegypti, as well as Culex quinquefasciatus just last year. People started discovering this in the mosquito species. And some of the results here are from Maturi et al. 2019. The bottom left shows a non-metric multidimensional scaling where it shows dissimilarity between different microbiome assemblages. And what's important to notice on this graph is the fact that the different treatment groups, like the newly emerged versus humans versus chicken versus rabbit, they're tightly clumped between each other, but they show differences or separation from each other. So within, they're more clumped and they are kind of separated. And that's really important to note because this shows that there are differences within the assemblages. And then if you look over to the right, you can see the kind of the proportions of the different bacteria that were seen in these treatment groups. The bottom one is the genus level. And you can see the variation that occurs between these, showing that this is very much true in mosquitoes as well. And all of these are really great studies showing this factor. We haven't necessarily been able to associate these changes with necessarily how it affects pathogen transmission. However, some studies do look at microbiome and their specific effect on pathogen transmission. One of the best well-known studied ones is Wolbachia, and it has been shown to decrease dengue, chikungunya, yellow fever, and Zika virus in Aedes aegypti. And although this is really well studied, it's not a naturally occurring bacteria in the Aedes aegypti mosquito, but there are other naturally occurring isolates that do exist in Aedes aegypti that have also had shown to have pathogenic effects. Some of them include Chromobacterium, Proteus species, and Panabacillus species, both showing decreased dengue in Aedes aegypti. And then we also have the opposite effect with Serratia odorifera, which both showed an increase of dengue 2 and chikungunya in Aedes aegypti. And although these are really great studies, they are also done under situations where they take the mosquito and they feed it antibiotic and they wipe the mosquito completely clean of its normal bacterial contents. And then they reintroduce a single isolate to see these effects. So really what our research is looking to do is kind of bridge these two areas together and looking at these natural changes that occur from feeding on different species and the microbiome changes and seeing if those effects create certain assemblages that can cause or change pathogen transmission in the mosquito. So our overall central hypothesis is that blood from different species will alter the mosquito microbiome, and this will be due to different physiochemical parameters, or like what's in the blood specifically, and that this will also influence arboviral dissemination within the mosquito. And below, it's just a theoretical framework of kind of what the research is about. So we have a Aedes aegypti mosquito that's in proximity to humans, which it predominantly feeds on, but they are known to feed on other species. And then it gets let's say it's uninfected and it takes a blood meal from a species, and this blood meal can cause microbiome changes. And Aedes aegypti are also known to take multiple blood meals within a single cycle of egg laying. So you can have multiple blood meals pretty close to one another so that they take a blood meal and you still have those microbiome changes when they take a second blood meal. And if that second blood meal is infected, they could be coming into contact with that changed environment. This could lead to selective pressures and changes in viral transmission in the mosquito. So there's two major aims to these studies. One is to examine the extent to which the blood meal source influences the Aedes aegypti microbiome. This will confirm as well as build on previous findings in the literature. We are going to use 5 different taxonomically diverse species to look at how these microbiome changes occur from this blood. And in addition to that, we're also going to look at what is in the blood to see if we can see any association between what's in the blood and these changes we're seeing in the microbiome. So a few of the factors that we've really honed in on a little bit more include like total protein, red blood cells, which also includes hemoglobin, as well as protein when it's digested, iron, as well as fats and lipids like cholesterol and triglyceride. And then our second aim is to measure the extent to which blood meal source and these microbiome changes affect viral dissemination with the Aedes aegypti mosquito. So we're going to use the same bloods as is done with the microbiome studies. And then we're going to use Symbis virus, which is a tocoviridae, an alpha virus, as a model system to assess these differences. So for the microbiome studies, the experimental flow goes like this. So we have an uninfected, unfed mosquito, and at 10 to 12 days old, we will feed it one of the 5 bloods. And at that point, we take some of the blood and we send it off for analysis to see what actually is in the blood. And then after 3 days, after feeding off the blood, which is enough time in which the microbiome is much higher, but all the blood has been digested in the mosquito, we then dissect out the midguts, we break them up, and we do a DNA extraction, and we send that off for high throughput sequencing using 16S ribosomal RNA, and that is a section of the genome that is conserved, highly conserved amongst bacteria, but also has hypervariable regions that allows us to hone in on the genus level of the bacteria that's in the microbiome. And then for the viral infections, we have 2 different treatment groups. They are both unfed and they are 10 to 12 days old. And then the bottom treatment group down here, they are fed a blood meal, an initial uninfected blood meal, and then we wait the 3 days. And then the other treatment group does not get an uninfected blood meal. So the one that gets first an uninfected blood meal is supposed to represent those microbial changes that are occurring. And then they both receive an infected blood meal with Symbis virus. And then at between 2 to 14 days, we sample every other day. And we collect the bodies, the legs, and the saliva to look at the infection, the dissemination rates or the spread of virus throughout the body, and then also the saliva to look for transmission potential. And then we take those samples and we get a virus titer on them through plaque assay on vero cells. And then what we're going to do is compare between treatment groups, treatment 1 and treatment 2, as well as between blood groups. We're going to look at the infection rates, dissemination rates, as well as the transmission potential, which are the titers in these 3 areas of the body, the rates as well as the titers. We're also going to look at the time of extrinsic incubation time to see if there's a difference. And then we're going to look at low versus high disseminators across all groups and see if there's certain microbiome assemblages that we are appreciating with these different levels of dissemination. So I have to be honest, the results at this point in time are pretty limited because we're still processing all the samples. We recently sent off a subsample of our microbiome work just to make sure that our DNA extraction is working properly. We are now doing a phenylchloroform extraction because we were having issues with some other extractions and they are single guts. So it can be a little bit tricky to get the sample properly processed. And good results, most of them came back as passing, so that's really good and we can go forward with those sequencings. And then we do have back a lot of the blood physiochemical parameters. So we have over 40 factors that we're looking at, but as I mentioned, we are looking at mostly honing in on those 5, and you can see a high variation between the different groups. So I'm excited to see if we can tie in some of those associations with each other with the microbiome that we're seeing. And then as for the viral dissemination, there's not much that we can actually glean from this at this point, as it's really early and there's a lot of gaps and we're still processing a lot of samples. But the top left chart is just the chicken blood samples. And you can see the chicken bodies are in the blue and you can see the viral titers. And then the chicken legs, we kind of started, all the bodies seem to be positive. So then we started just focusing on the legs so that we could get an idea to guide our future research to see, okay, when can we start processing them or start taking samples, and are they all disseminated by a certain date so we don't have to continue getting samples. And And you can see that the legs are, at least on day 10, are a little bit lower. And then you have the bovine blood together on the right. And then I grouped the body dissemination rates on the bottom left. And you can see that the equine blood, the titers are a bit lower in the body on day 2. That's the only day at this point in time that we have together. But the numbers are so small at this time, it's really hard to glean anything from this. And then when I put the legs together, you can see on day 12 that they actually look pretty similar at this point at day 12 for titers in the legs. But definitely need to do more in this, and we're processing samples. I stained another 40 this weekend, so I hope to add that data and continue rolling with those. And so overall, the major implications of this study could be, we're looking at a possible additional factor of this vector pathogen host interaction that could lead to possible emergence in populations if certain species, or if a mosquito bites off of a certain species and it increases transmission, what could that actually mean for, in the field? And this is also an aspect or an impact of land use change or species assemblages. If species are changed to something that is not normal in an area, and mosquitoes come in more contact with those animals and bite off of them, what does that do for transmission of viruses or pathogens in general? It could also majorly change how we perform arboviral research. So if we're seeing changes in the types of blood that we feed and the viral, the results that we're getting, there really needs to be more of a set standard for how we perform it if we're trying to compare different research. And then further directions would be to do additional viruses and see if it's true for others, if this does end up being, if it is a proof of concept. And then also, as I mentioned, looking for possible novel bacteria to look in viral control or pathogen control. If we see certain assemblages or certain isolates really popping out in those high versus low disseminators, this could let us hone in a little bit more on maybe naturally occurring isolates that could be used for further viral control or pathogen control in nature. And that is it.
Okay, thank you, Melissa. Any questions for Melissa?
Hey, thank you, Melissa. I have some questions. So how many, you said that you already had the blood physiochemical samples analyzed. How many samples did you run through?
So I only ran them on the ones that I actually fed the mosquitoes. So that was the whole point. So what do you mean? Oh, all 5 of them. So
no, how many different blood samples did you have?
So actually, yeah, no, that's a really good question. We're only doing the ones that I did the microbiome samples on. So if I were to feed, so blood varies, you can have it anywhere from, there's always a range between norms. I think that's what you're going at. There's an average and then there's a range, but you can't necessarily go on the range when you're feeding the blood. So it's only on the blood that I fed those mosquitoes. And then I'm going to get what that comes out as, if that makes sense.
I understand. I'm just asking how many samples you looked at. Did you have 2 samples of a chicken? Did you have 3 samples of a chicken?
No, it's 1 sample, but they are grouped. And then I did get them for our viral infections as well if we needed them, but we haven't sent them off. Because if I'm not necessarily I think I know what you're getting at, but if I'm not necessarily going to be feeding that blood to them and then getting that microbiome data, I don't necessarily want to put it into my assessment because there is that variation that can occur.
Right. Do you think that it's better to have the variation rather than a single sample?
No, not for this specifically. I understand why that would be. But if I do like multiple replicates, so if I repeat the experiment, yes, that would be ideal. But to just send off samples of like chicken blood and see what their values are, I can go to the literature for that. I don't necessarily need to send my samples off for assessment if I'm not actually going to take the microbiome samples from them. But if doing multiple replicates, yeah, that would be ideal, definitely.
Okay.
On different days, yes, that would be ideal.
Also, I had a question on, So you said that you're looking at mosquitoes that are 10 to 12 days old. Why did
Yeah.
you select 10 to 12 days old mosquitoes?
Yeah, so usually it's a little bit younger, and that was off of the guidance of the lab to try to get higher feeding rates. So it is pretty difficult, especially their fourth generation out is when I start. And earlier on, we were seeing lower feeding rates when they were younger, so we decided to push it back a little bit.
Okay. Are you concerned that you're going to have 29-day-old mosquitoes towards the end of your experiment and you might run out of samples?
Yep. So we've ran all the way through day 14. It does start to teeter off. I do have to, especially for those refeeds, the refeeding rates. So my initial feeding rates are pretty good, 80 or higher. The refeeding rates, depending on the species, can definitely be lower, which is a difficulty, definitely a hardship of this study, especially with younger. I know I've talked to some other people in the research, and they have highly laboratory raised, like high generation laboratory raised, and their mosquitoes readily refeed very easily between 1 to 3 days, but ours do not. And I don't know if it's the system we're using or if it's because they are so, they're only 4th generation out. But yeah, no, it is something that when it originally happened, we were like, they're going to be a month old by the time that we're getting our finished viral product. It is.
These are aegypti, right?
Yep.
So are you starving them before you feed them?
We're doing all of it.
All right. And then finally, why did you select Synbis virus?
So first of all, the reason why we picked it is because it is a BSL-2 agent, and we wanted to start with that one. It's also highly used as an option for kind of a model system. So a lot of research is done with this virus, especially previously, to use it as a model system to see different physiological changes and how arboviral transmission can change. It is not normally transmitted by Aedes aegypti at all, but they are permissible and susceptible to infection. So that is one way in which we can look at this and do a proof of concept before we start moving to other viruses to assess it.
Do you find Synbis in all 5 of the different organisms that whose blood you're checking?
No, it's mostly a bird or an avian pathogen. It can be found in others though, but no, it is mostly an avian, definitely.
Okay.
And
Thank
again,
you.
it's just a proof of concept. It's just like any other viruses. We feed bovine blood for dengue. Like a lot of the time we don't use the blood. We don't use human blood in all our studies. Yeah. Thanks for the questions.
Mm-hmm.
Any other questions out there?
Well, I have one curious. I have one question that is more my curiosity, and another one that I was thinking also. So usually they pick up like the bacteria from where? From the sugar sources?
Yeah, anywhere. So it's the water environment, but it also changes throughout their life cycle. So larvae have a different bacterial composition than pupae. They're also feeding when they're larvae. They're not feeding when they're pupae. And then when they emerge, when they go out and they are getting bacteria from like the nectar that they're drinking as well as blood. So it gets put in there, and it's definitely from the environment. And that's mostly appreciated from the fact that we do see different microbiomes from same species, even from different geographic locations. And they have found also, if you take those mosquitoes from different geographic locations and you bring them into the lab and you breed them for like 3 to 4 generations, they actually get the same bacterial assemblages. So it is mostly from the environment that they're living in and they're raised in.
Okay, yep. And the other thing that I was thinking is, like you mentioned that you are going to measure like different parameters in the blood.
Mm-hmm.
And I was like remembering I used to, well, in the bacterial laboratory, when you have to diagnose and like to tell which bacteria it is, sometimes you use like these little things that they have like a miniature test of physiology and biochemistry, and you can tell apart between bacteria sometimes based on like the type of amino acid that they use or the type of like sugar that they ferment, or different like very specific chemicals. So are you going to evaluate like more deep those like type of parameters, like instead of like total protein,
Oh, yeah,
like
we
more
have,
amino acids or things like
oh
that?
yeah, we have everything. So we have white blood cells, neutrophils, basophils, different like creatinine, blood urea nitrogen, like all of the like over 40 of them. But these are the big ones that we kind of honed in because of looking through the literature. So protein and iron are believed to be associated possibly with the increase of the bacteria that we see. And then also I wanted to do glucose as well, of course, because we do see it from that. But the issue is with the additive that you add in order to keep the blood really stable and really fresh, or as similar to what it is going to be in nature, you add dextrose to that. Because like, as you know, the red blood cells eat that glucose really quick in the blood. So if you get the blood, it's not going to have any glucose in it at all. So we wanted to keep that. So it's really high. So it doesn't really work very well to look at that because it's so extremely high. But yeah, we have so many different ones that we're going to look at. But those were the ones that we kind of focused in on just because of what we were seeing in the literature and things that we thought could possibly have the most effect based on digestion.
Thank you.
All right.
Should continue.
Okay. Sorry, I'm trying to stop sharing. There we go.
Bob, you're up, right? See the screen.
I'm sorry, I'm having trouble trying to find some of the controls.
No,
on
we can hear you and we can see your screen. So that's
Okay,
the most
great.
important.
All right, let me, there we go. Is it in presentation mode? Yeah, there we go. Okay, so yes, hello, thank you. My name is Bob Aldridge, and I'm presenting on some of the PhD research that I'm working on in Dr. Alto's lab. It's looking at the influence of insecticides and its influence on Aedes aegypti when it's either infected with a pathogen or if a mosquito is exposed to a pesticide and then later becomes infected with a pathogen. So in this case, it's dengue is the pathogen, and then a number of different insecticides that we're interested in. So part of the problem, just looking at the background of this, is that we use quite a number of chemical controls in trying to control mosquitoes, and that can overwhelm an environment. We might be overstimulating an area, or not putting in enough. So a lowered application rate may have some deleterious effects on vector control, in that if we're compromising the mosquito while it's growing, it may then become more susceptible to pathogen infection once it's able to make it to the adult stage. Sorry, I had a couple things coming up. Right, so just as I said, insufficient application, insecticide resistance, increased potential of exposure to sublethal dose and concentration. These are all background issues that concern part of the project that I'm interested in. And then I hate this, I can't see some of the, I can't see some of the top of what I'm saying. So here are the questions and concerns that I'm interested in. What effects do sublethal concentrations of insecticides have on vectors? And does exposure to a pathogen make the vector more sensitive to insecticide? And does exposure to insecticides make the vector alter its susceptibility to pathogens? And so our hypotheses then are very similar to that. Insecticides could then alter vectorial behavior and physiology, or exposure to a pathogen may alter vector sensitivity to insecticides. And finally, insecticides could influence vector competence. Getting to the meatier portion of this, so our organisms are Aedes aegypti, which is a very common mosquito. It's found globally. It's difficult to manage. It's able to transmit a wide range of pathogens, including dengue, chikungunya, Zika, as well as yellow fever. We are using the susceptible strain, or the Orlando colony, and that is a colony that's maintained at the USDA as well as in Dr. Alto's lab. It's been maintained at the USDA since at least 1952, if not later, or I should say earlier. And I'm looking at both larvae as well as adults. So the pathogen I'm interested in is dengue virus serocomplex 1. There are multiple serotypes, and right now dengue virus infects over 300 million people around the world. And there are some hemorrhagic and shock syndromes associated with dengue. The sublethal insecticides, so a sublethal concentration is considered a dose less than 25, where 25% would die. I'm interested in looking at formulated products, and these include things like permethrin, methoprene, BTI, which is short for Bacillus thuringiensis israelensis, and spinosad. So these three combine to form kind of a complex structure that interact with one another. And specifically in objective 1, I will be identifying the sublethal exposure that generates a dose that can kill 25% of the population. This will be looking at a series of different doses, increasing in concentration after exposure to identify the optimal dose range. So basically, this is a small breakdown of what I'll be doing. I'll look at different concentrations, expose larvae to those concentrations. Shortly thereafter, there will be adults that will emerge and larvae that will die. Following that, I can chart how many emerge and generate a lethal concentration at 25 or 50%. And in this case, we're going to be using 50%, the LC50, because we wanted to make sure that the effect of the pesticide was evident. Earlier studies or earlier proposals looking at the 25, LC25, but we kind of erred on the side of caution and moved it up to the LC50. So yes, we were able to identify an LC50 for several pesticides. Specifically, we looked at Vectobac. Vectobac 12AS is a BTI, Bacillus thuringiensis israelensis, and I have identified dose curves here, and also using that dose curve, I've been able to identify the optimal LC50 that I can feed to these mosquitoes, these mosquito larvae, and generate adults that were exposed to that. So we have the BTI, we have Nigard, which is actually a pyroproxyphene. If you look at the bottom, this is in nanograms of Nigard, which is an insanely small amount of pesticide. Here we have Altocid, which is the active ingredient is methoprene. So we were able to identify that one. This is abate, or Temifos is the active ingredient. And actually, Temifos has been discontinued in the European Union, so they're no longer using it. It's starting to phase out in certain locations. However, it is still very practical to use, and it has been approved by the WHO for use in drinking water. So this is an organophosphate. The other ones that we looked at, so let me back up here for a second. This is a bacteria that utilizes proteins that then form holes in the midgut of the larvae. And those holes then disrupt the homeostasis of the developing larvae, and then it dies from that disruption. Nigard uses pyroproxyphene. Pyroproxyphene is a juvenile hormone mimic, so that instead of it molting and behaving in the proper set of series of behaviors, this juvenile hormone mimic tricks it. And while it's in its larval to pupal stage, specifically it acts as a pupicide. So as it's molting from pupae to adult, it dies. Alticid is methoprene. Methoprene is also a juvenile hormone mimic. So just as similar to pyroperoxyfen, as it's molting from one stage to another, it dies. And this one is particularly sensitive to larvae. So as larvae are maturing from larva to the adult to the pupal stage, they die there. Abate, like I said, is an organophosphate, so that acts on disrupting the axon channels. So I should say the sodium channels of the nerve. And then finally we have, not finally, but we also have natular. Also, the active ingredient is spinosad. So this one is is considered an ecologically friendly one because it's derived from a bacteria rather than one that's synthetically created. This one is very similar to the organophosphate temophos, except this one influences the acetylcholinesterase enzyme channels, so it prevents it from actually breaking down the acetylcholinesterase. I do also have Vectolex, or I'm working on it, but I don't have a full graph of that. I don't have the full data for that. It's actually Vectolex is Bacillus sphaericus, and so it's much more effective with Culex quincofasciatus than it is Aedes aegypti. So the amount needed for it to actually be active on Aedes aegypti is much, much higher. So instead of actually having data for that, I will give you a mosquito with a hat. Okay, so the next thing is after we generate these LC50s that we're able to feed to the mosquitoes, we want to be able to feed pathogens to the mosquitoes. And in doing so, we're going to culture cells, and once they become convalescent, we will infect them with a particular pathogen, in this case dengue. Then we will infect the blood. We'll take those cells, add them to blood, right? Those cells and bacteria then, I'm sorry, not bacteria, but those cells and viruses then are infectious in the blood. We will then feed that blood to mosquitoes. The mosquitoes will then be dissected, their legs dissected. They will also be used as a capillary action. We will grind up their bodies, and all these samples, legs, body, and saliva, will be analyzed using PCR. So far, I've been able to run some of these studies, but this is where that information becomes critical. We take the LC50, expose the mosquitoes to that, analyze it, and then we run through the how much, like what impact these different exposures might have to the ideal extrinsic incubation period of these mosquitoes. So we do have some results before COVID locked everything down. We have, I had 846 samples of bodies. Unfortunately, we go bodies, legs to saliva. So I have all of the bodies ground up, PCR'd, and everything. That's across 3 different replicates, but only looking at 3 different pesticides and control. You'll notice that this is the proportion infected from those samples. Each set of columns is a particular pesticide or treatment. And then within that set of columns, it's divided as the first column, 3-day-old post-exposure, 5-day-old, 7, 10, and 14 days old. Now, I've got some wonky results. I think that I need to do a little closer investigation of some of the data. But so far, it just needs to be looked at a little closer because some of the data is just wonky. Like it's very nice. The abate has this expected increase in number of infected or proportion infected. However, at day 10 and then day 14, they just completely collapse. So everything so far at day 10 and day 14 has been collapsing. I'm not exactly sure why or if that's a normal thing. So I need to converse with Barry and see if some of these results are normal or if they're not. Moving on, I will be looking at the legs and saliva once I'm able to get back down to FMEL and run some PCR. The final step in this analysis is going to be exposing adult mosquitoes once we're able to generate an LC50 for adults, looking at after they've been exposed to a pesticide, if they are then more or less sensitive to a pathogen, right? So first we get the LD50 for the sublethal exposure of Aedes aegypti, looking at least 2 different pesticides. This would be a permethrin and then an organophosphate, because there aren't many adulticides that they make anymore for chemical control, or different modes of action, I should say. Look at the extrinsic incubation period under the control and how long it takes after exposure for the insecticide to influence the extrinsic incubation period, and then determine if prior dengue exposure does influence that susceptibility. So very similar, we will be generating some infectious blood by infecting some cells, feeding those viruses to mosquitoes, and then after the ideal infectious time, ideally like when they would be able to bite and then transmit a pathogen, expose them to a pesticide and see if that exposure changes their susceptibility. Their exposure to the pathogen changes their susceptibility to the pesticide. So these are some expected results, right? Days past exposure, possibly there will be a change in susceptibility. Their proportion mortality may increase or decrease, I'm not exactly sure which. But that's ideally what we're looking for, and therefore the kind of interaction of these 3. Just to recap, this is a big problem because not everywhere is exposing or applying pesticide at the uniform rate. And so if we're not applying at the ideal or uniform rate, then we're either under or overexposing, and that can therefore alter potential susceptibility to pathogens or alter pathogen susceptibility for insecticide in these vectors. And with that, I would like to thank the members of my committee, and also, of course, members of each one of these labs and their support. So I very much appreciate that. These are some references cited that I mentioned. And with that, I will take questions or comments. I can't see anyone.
Some comments for Bob out there.
Thank you.
So for the so you said you ground up the bodies. Do you plan to like ground up just the head and thorax to capture the the parasite virus that are kind of past the midgut level instead of whole body?
No, everything has been ground up. So the head, thorax, and abdomen have been ground. Are there any other questions?
Okay.
Great.
I think that, thank you, Bob. And I think that concludes our update for current research in my lab. Is any other final comments? Otherwise, we'll log off and we'll see you guys at a meeting in December.